Hello everyone, Mr. Walker here. In the last lesson we did talk about sensory receptors, talked a little bit about the specific ones, mechanoreceptors for touch, and a couple of the chemoreceptors for the sensations of taste and smell. This one here, we're talking about the photoreceptors. Whenever you see the prefix here, photo, what we are talking about is light. So these are receptors that are very, very specific for capturing light energy. But keep in mind, once they do capture that light energy, the message does have to be converted into an electrochemical potential before that message is sent to the brain. Picture that we do see here, this in fact is showing, well, not our eyes, but this is showing a compound eye like you would find in an insect. Kind of interesting, but in fact completely different from the camera style eye that we have in our eyes and that other mammals have as well. Before we take a look at a picture of the camera style eye, realize as well that probably, well, most animals, if not all animals and all living organisms, probably have at least some ability to detect light, not necessarily forming an image like we do or like this compound eye does, but at least be able to tell the difference between nighttime and daytime. So this here is the camera style eye and various different structures that you do need to know and the function of various different structures. So what we do have are three different layers. I'll just label them here as 1, 2, this red flap, and 3, this yellowish flap that we see here. Light comes in through some structures at the front of the eye. Obviously, these have to be clear structures for light to pass through. So light is going to pass through all of these different structures. All of these need to be clear until it does reach the back portion of the eye, the innermost layer of the eye that I'm circling here. And this innermost layer of the eye is referred to as the retina, and it is on the retina that you will find the photoreceptor cells that are going to be capturing light energy. So we'll come back to this picture and we'll take a look at this picture a couple of times. In the eye itself, it is in fact an extension of the brain. So there is some rudimentary processing that does take place in the eye itself, but ultimately to perceive and process that image, the message does have to go to the brain and it does travel through this nerve right here, which is the optic nerve, and from there it's going to go to the occipital lobe of the brain, and that's where it's going to be processed. Details that you need to know about the retina: there are two different types of photoreceptors. You do need to know specifically a few different things. You need to know their names. They are the rods and the cones. Down at the bottom, there are a couple of representations of these two different kinds of photoreceptors. The rods get their name from the fact that they are, for the most part, sort of a rod shape, and the cones from their structure, which is a cone or conical shape. So for the rods and cones, you do need to know where they are located within the retina. You need to know about their light sensitivity, and you need to know specifically what they are for. So for the rods, these ones here are very light sensitive. Very, very small amounts of light are sufficient to activate these photoreceptors. What they don't do, though, is they do not distinguish between different colors. So the rods are good for black and white and shades of gray, but not for processing any kind of color. That is what the cones are for. So the cones are not as sensitive as the rods. They do need brighter light in order for them to be activated, so not as sensitive, but they are in fact for colors. Different organisms have a different number of cones. As we will see in our eyes, there are 3 different cones, and there is just the one type of rod. In humans, most of the cones, they are located in the fovea or the Fovea Centralis. Where we find most of the rods being located is actually not in the Fovea Centralis as well, but it is around the periphery of the retina. If we go back to the previous picture that we had here that is showing the retina, again, it is the innermost shaded in yellow flap or layer that we see here that is the retina. When light does come directly in through the eye, where it would strike is this region right here, and that region is referred to as the fovea or the fovea centralis. So it is in that location there that we do find the vast majority of the cones that again are activated with very bright light. Where we find the rods are going to be, well, actually not in the fovea at all, but if we go away from the fovea in any direction, then what we do have is an increased number of the rods, which are responsible again for the black and white vision. The graph that it's showing here at the lower left-hand side, what it has along the x-axis is the distance away from the fovea. So this in fact is the fovea right in the center here. And as we go further away from the fovea in either direction, that would then be the periphery of the retina. What it's plotting along the y-axis is the density of the photoreceptors. So again, of the two different kinds of photoreceptors that we have, where we have the highest concentration or density of the cones is in fact right at the fovea. So very, very high density there. What happens if you go away from the fovea? Well, we can see that the density drops off very, very quickly. So very, very few cones that we find outside of the fovea. In the case of the rods, exactly the opposite. For the rods, there are none of them right in the fovea, but as we go away from the fovea, we can see that there are higher concentrations the further away we get. This picture here is looking into the eye itself. So a doctor using a light, looking through the various transparent structures at the front of the eye. So looking through the cornea, looking through the pupil, through the fluid layers or fluid components at the front and the back of the eye, the aqueous and the vitreous humor respectively. This is what the doctor is looking at, and what we do see here, as it says, is the retina itself. All of these squiggly lines that we see here, these are blood vessels that are running through the back of the eye, supplying oxygen and nutrients to the retinal cells themselves. It does also identify here the most sensitive part of the eye, the highest concentration again of the cones in this area. And it also identifies something else. It identifies the optic nerve. So this is where the optic nerve exits out of the back of the eye. Information coming from the retina, it's all going toward that optic nerve. All of the information from sensory neurons going toward the optic nerve and then exiting out of the optic nerve, going back toward the occipital lobe of the brain. This location right in the middle here, though, there are actually no photoreceptors that are located. There are no rods or cones located where the optic nerve exits the eye, and for that reason, it's also referred to as the blind spot. If light does strike in that location, it's not going to activate any photoreceptors because there are no photoreceptors located there. This picture here is showing us what is called a detached retina. So if we again take a look at the three different layers, this is the outermost layer. This one right here, number 2, is the middle layer, and the innermost layer, which again is the retina, this is the one that has pulled away from, it should be adjacent to, but it's pulled away from the middle layer, and that middle layer is called the choroid layer. It is pulled away, and what they're trying to show here as well is that fluid at the back of the eye, the vitreous humor, it's getting in between those two layers, and it's pushing those two layers away. And that again is a detached retina. For all of these photoreceptors, what you do have to have is something that is going to capture light energy, and those are what are referred to as pigments. So these pigment light absorbing molecules, what it is a retinal, which is really vitamin A. And this is one of the reasons for having vitamin A in your diet, is for the proper functioning of your eyes, and also a protein that is called opsin. Rods are the ones that we primarily talk about in terms of the light-absorbing pigments and the chemistry involved. And the light-absorbing pigment in rods, it is referred to as rhodopsin. As we will see, when light does strike the rhodopsin, there is a change, an actual physical change in the structure of the rhodopsin, and that's going to lead to a whole series of events, ultimately leading into a message reaching the brain. In humans, I mentioned that there are three different cones. In these three different cones, there are three different pigments that absorb different wavelengths of light. So you have one of the cones, which is for blue light. You have another set of cones, which are for green light. And the third type of cone, they are for the red light. You certainly don't need to know these percentages, but these ones here, which make up, oh, well, it is about 98% of the photoreceptors, these are the ones that are located in the fovea itself. So these ones are located in the fovea. So I said that most of the cones are located in the fovea. The exception are these blue ones. Blue ones are actually located outside of the, I may have said retina, but it's outside of the fovea, where you do find the blue cones. So this allows us to detect at least the full range of what we refer to as the different colors. There are some other organisms that have a fewer number of cones. They have a more limited color vision, and there are some that have even more than the three cones that we have. In birds, for example, they're able to see into the ultraviolet range. They have four different cones. In some insects, they are able to see into the infrared range, and they possibly have four cones as well that allow them to do that. This picture here is showing some of the enlarged details of one of the rods. And in the rods, what we see here is something that is called a rhodopsin disc. And it's in these discs that they're using these purple circles to represent the pigments. So it's these pigments again that are absorbing the light energy. When that light energy is absorbed, this is showing that retinol, the vitamin A. I mentioned there's a structural change. This is the structural change that does take place when light does strike the photopigment, in this case the rhodopsin. So some details in terms of the chemistry of what is going on. It's a little bit backwards compared to what we've been talking about so far. In this case, with the photoreceptors, we would expect sodium channels to be closed in the dark, but in fact, the sodium channels are open in the dark. But what those neurotransmitter or what those photoreceptors then do is they release a neurotransmitter, which just happens to be inhibitory. So that means that there are fewer action potentials going to the brain. When we do have the rhodopsin that is split, now the photoreceptor channels are going to close, and when it's going to split is when those photoreceptors are exposed to light. So in this case, what's going to happen is the neuron inhibition is going to be removed, and that's going to allow for action potentials to be sent to the brain. Let's take a look at a couple of pictures here that maybe explain this a little bit more than all of that text. We're taking a look at, on the left-hand side here, our rods that are in the dark, and we can see here that the sodium channels, they are open in the dark. What does that mean? Well, at the end here, this message is going to have to be sent on to a sensory neuron, so we will have to have a neurotransmitter. But in the dark, sodium channels open leads to the release of the inhibitory neurotransmitter, which does mean that there are fewer action potentials sent to the brain. So even though in the dark, we could say that the photoreceptor is stimulated because sodium channels are open, sensory neurons will decrease the number or rate of action potentials that are going to the brain. What happens when, well, the lights are turned on? Well, now we're going to have a change in the shape of the rhodopsin. That's going to lead to the closure of the sodium channels, which means the neurotransmitter that's now being released from the end of the rod is going to decrease, but that was an inhibitory neurotransmitter. So less inhibitory neurotransmitter means that now we will have more action potentials or an increased rate of action potentials that are traveling through the optic nerve and going to the brain. Same thing here. Here it's actually showing where we do have the neurotransmitter that's being released in the dark. Again, the neurotransmitter, this one is going to be inhibitory, so the number of action potentials that are going to the brain is going to then decrease. At least that would be the typical situation. In the light, we have less of the inhibitory or no inhibitory neurotransmitter, so now we would have an increase in the rate of action potentials going toward the brain. And this picture here is showing where we do have our initial stimulus. So here the light is coming in from the left, so it's traveling through several layers of cells that we'll talk about in a minute to eventually reach the photoreceptors. So in the light, what we're going to have is going to be hyperpolarization, but the tricky part is that does lead to depolarization in the next adjacent cells, in this case the bipolar cells, ganglion cells, and eventually sensory neurons going to the brain. And we can see that in the light, yes, we do in fact have an increase in the rate of action potentials. So back to these several layers of cells. You don't need to know these individual layers of cells, but it's kind of an interesting story. The picture that we're taking a look at here, if we take a look at the picture at the upper left-hand side. First of all, what it's showing is that the back of the brain, way over at the right-hand side, is where we do find the photoreceptors. So it identifies the choroid layer, which means, of course, a little bit further back still would be the much thicker outer layer, which is the sclera. The sclera. But the key here is take a look at where the photoreceptors are located. Whether it's the rods or the cones, they're adjacent to the choroid layer, but then in front of, toward the interior of the eye, we have several layers of these cells. And light is coming from the left. So light has to pass through several layers of cells before it even reaches the photoreceptor. So it doesn't make a lot of intuitive sense to put those cells in front of the photoreceptors. Not only that, once these photoreceptors generate a message, that message is actually traveling out through the horizontal cells, the bipolar cells, and eventually to the sensory neurons, which is going to take the message back in this direction. So really kind of a backwards design, not really the best engineering in terms of the mammalian eye. These ones here, I'll let you read over this information, but just to give you kind of the highlights, this picture here is showing that we have the red color in a rows and we have the green color. Whether you see the red as being brighter or the green as being brighter kind of depends upon how bright the light is. In bright light, you will see the red a little bit better. In dim light, you will see the green a little bit better because the green actually does activate the rods more than the red activates the rods. The one in the middle, a little bit of a picture showing instrumentation, either in a ship or in a plane, and it's showing that the instrumentation is not white in color, it's red in color. And the reason for that is that the captain or the pilot, they do want to have night vision, which means they do not want to be exposed to bright light or they lose that night vision right away. If there is red light though, the red light doesn't affect the rods, so they can take a look at a panel with red instrumentation and it doesn't affect their ability to see outside of the window, whether it is a ship or a plane, and see what's going on outside of the vessel. And the last one here, the picture at the bottom, taking a look at the three different colors, the red and green around the outside, the blue toward the center. And the idea behind this is that it's difficult to see all three of those colors clearly at the same time. And part of it does have to do with the location of the blue cones being outside of the fovea. And it also has to do with the fact that the blue light is bent a little bit differently, refracted a little bit differently than the red and the green light, and that's why it makes it difficult to see all three of those colors crystal clear at the same time. Anyway, just some interesting facts. This one here kind of neat as well. This one is showing the eye of a fish that you can see here, where it is reflecting any incoming light. And in fact, that's the case that you see with many nocturnal animals as well. If you're driving in the dark and you see eyes to the side of the road, that's because these animals do have on the choroid layer a reflective surface that allows them to see a little bit better in the nighttime. And that is not something that we would have in our eyes. Little bit on the optics of vision, so some good physics here. Humans and other mammals, we do focus the light by bending the light. So when we talk about focusing, it is synonymous with bending or the term refraction. In order to focus the light, you do need to bend and refract the light. This takes place using two different structures in your eye. Right at the front here, this clear window is not only a clear window to allow light to pass through, it is also bending and focusing the light. This is called the cornea, and in some cases it's referred to as the fixed lens. Fixed in the sense that you can't actually change it. The other structure that focuses the light, this one right here, is the lens. And the lens, again, bends and focuses the light. But this one we do have some control over, although of course it is autonomic control over it. By changing the shape of the lens, that is a term that is referred to as accommodation. And what is changing the shape of the lens are some muscles that are located around the lens itself, and these muscles are referred to as ciliary muscles. So what this picture here is showing is that when we are viewing an object up close for near vision, so this dot here is representing an object that is close, we do have to have significant accommodation that is taking place. The ciliary muscles are going to be contracting, and what they're going to be doing is they're going to be making the lens round, and they're going to be making the lens, well, kind of fat. And that allows it to bend the light. The idea behind bending the light is we do want these light rays, when they're coming in, to converge right at that point where we have the retina. If they don't converge at that point, then the object will not be in focus. For objects that are viewed far away, you don't need to have that same degree of accommodation. In this case, instead of the lens being fat, the lens is going to be, well, it's going to be thin. You don't need to bend the light as much. So now the ciliary muscles, instead of having to contract and work really hard, they are going to be relaxed. So the lens, yeah, I said thinner. Here it says flatter. The other side, I said fat. Here they say thicker or rounder in terms of its shape. So two different conditions. The first one, nearsightedness. So nearsightedness means that an individual can see near objects fairly clearly, but they have difficulty viewing objects that are far away. So the problem when an object is far away, if we take a look at this picture, so this is representing something that is far away. I mentioned that what you want to have are the lines converging on the retina, but in this picture at the top, clearly they do not converge on the retina. They converge way in front of the retina. What we want is what we see at the bottom, where now they are converging right at the retina. So what's the problem if someone is nearsighted? Well, no, they cannot see objects that are far away, at least not that well. The problem is that, well, possibly the eyeball is actually too long. So eyes don't necessarily have to be completely spherical like a ball. They can be shorter from front to back. They can be longer from front to back. And much of this is dictated by the shape of the skull itself and the orbits the eyes are sitting in. The other possibility is that the lens could be too round. Again, if it's too round, then what we have is more bending or more refraction of the light. And in this case, it's actually bending it too much if the lens is too round. So what happens? Well, the images of near objects, they do fall on the retina. All of that is okay, but those of distant objects, it's as if they come into focus in front of the retina, and by the time they do reach the retina, they would then be out of focus. What do you do to correct this? A concave lens. Concave is a lens that curves in. So if we have a biconcave lens, it's something that would look like this. This is also referred to as a divergent lens. So if we do have light rays that are coming in, it bends them further apart. That would be divergent. And that, in fact, is what you need to do for an individual that has nearsightedness. The light is being bent too much. You want to bend the light less. And the way that is accomplished is by sticking in front of the eye a concave lens. For farsightedness, really exactly the opposite. In this case here, individuals are able to see objects that are far away but have difficulty with objects that are close to them. In terms of the eyeball shape, too short rather than too long, and the lens too flat. It doesn't bend the light enough. So distant objects, they are in focus on the retina, but objects that are viewed up close, so in the picture here, this would be representing an object that is too close. And again, we can see that the light rays, they don't converge right on the retina. If these light rays could pass through, they would actually converge behind the retina. Of course, that doesn't happen, and that means that the object would be out of focus. This one here is corrected for by using a convex lens. Convex lens, exactly the opposite shape as the concave lens. And this one here, when the light is coming in, it's going to bend it in this direction. So the concave was called a divergent lens. This one is a convergent lens because these lines are converging, and in this case they're converging right where we want them to, which is on the retina itself. And final slide here, just a little bit about the eye structure and the evolution of the eye. It's often argued that it doesn't make sense that evolution created something as detailed and complex as the camera style eye, because if you have only a partial eye, then it's of no use whatsoever. Well, not necessarily the case. If you're an organism that just simply needs to be able to tell when it is light and when it is dark, all you really need are photoreceptors. We can have some organisms that have all of these intermediate forms. So these ones here aren't necessarily going to be forming an image like we form an image, but they can still not only detect light, but now they can tell the direction that light is coming from. This one here, not quite as complex as our eye, so again, this is going to be a fairly primitive eye, but we do have a lens here, perhaps some sort of image that is being formed. So we have these various different stages that they do think that probably evolution took us through in the formation of our much more complex camera style eye that we have today. So remember that evolution, it's not like one big step, but it's a whole bunch of tinkering along the way, small changes over very long periods of time, millions and millions of years, tens and hundreds of millions of years, eventually to get to the very, very complex structures that we do have in our body, including this camera style eye. Keep in mind as well, though, we did say that the camera style eye, not necessarily the best design, but this is what evolution did come up with, and clearly it's something that does work for us.